welcome back to my YouTube channel, and in today's video we're gonna look at the urea cycle. My name is Vernon Nsofwa. But before we go further, as always, if you're new to my YouTube channel, go and hit that subscription button, and always don't forget to like and share. I would really appreciate that. Please go down and subscribe. Let us get started. First of all, we need to define what the urea cycle is. So the urea cycle, this is simply the process that is taking place in the liver cells. And in this process, we are converting this toxic substance that we call ammonium to a less toxic substance that we call urea. So you are going to see how urea is being produced from ammonium. So this process is taking place in the mitochondria as well as in the cytosol of the liver cells. Okay, so you can see that what we have here, this is the mitochondria, and this is the mitochondria of the liver cells. But you can see that part of the process is outside the mitochondria. So it is taking place both in the mitochondria of the liver cells as well as outside the mitochondria of the liver cells or in the cytoplasm. Okay. Now, let us see what's involved in the urea cycle. What are the steps that are involved in the urea cycle? So, the first step, we have got our ammonium, and then we have got bicarbonate and 2-ATP. So, in the first step, we're using ammonium, bicarbonate, and 2-ATP. And what you should know that this ammonium is coming from amino acid metabolism. So, when you are breaking down amino acids through deamination, we are going to produce ammonia. And ammonia is going to be converted to ammonium. You should understand the difference between ammonia and ammonium. So this is ammonium. It's coming from ammonia. And ammonia is coming from deamination of amino acids. So in the first step, we're going to add ammonium plus bicarbonate plus 2-ATP. And we're going to form this compound, which we call carbamyl phosphate. So carbamyl phosphate is coming from these three substances. The enzyme here is what we call carbamyl phosphate synthetase 1. And this enzyme is very important because this is the most regulated enzyme in this cycle. So this is the regulatory step. Now, what's going to regulate this enzyme? So here we have got this compound that we call N-acetylglutamate. N-acetylglutamate is going to activate CPS1 enzyme. So N-acetylglutamate, this is a compound that is made from acetyl-CoA and glutamate by an enzyme that we call N-acetylglutamate synthetase. So this enzyme here, N-acetylglutamate synthetase, it's combining acetyl-CoA with glutamate to produce N-acetylglutamate. N-acetylglutamate, when it is produced, is going to activate CPS1 enzyme. And CPS1 enzyme, when it's activated, it's going to convert ammonium, bicarbonate, and 2-ATP to carbamyl phosphate. And this is very important step. The regulation of this enzyme depends on the absence or presence of this compound. So when this is available, the enzyme is going to be activated. When this is not there, the enzyme is going to be inhibited. Now, let us see what's happening in the second step of urea cycle. So in the second step of urea cycle, we are going to convert carbamyl phosphate to citrulline. But how are we converting carbamyl phosphate to citrulline? So outside here in the cytoplasm, we have got this amino acid that we are calling ornithine. So ornithine is going to enter the mitochondria through the ornithine transporter 1. And when ornithine enters the mitochondria, it's going to react with carbamyl phosphate. So ornithine will react with carbamyl phosphate to form what we call citrulline. The enzyme that's catalyzing this reaction is called ornithine transcarbamylase. So ornithine transcarbamylase enzyme, this is an enzyme that is going to catalyze the reaction between ornithine and carbamyl phosphate so that we can form citrulline. Now, what will happen to citrulline? Citrulline is going to be transported to the outside of the mitochondria. And here it's going to be converted to what we call adeninosuccinate. But what's going to happen here is that there is this protein that is found in the membrane, this transporter protein. So this protein is called citrulline. So citrulline, what is it doing? It's going to transport aspartate from the mitochondria to the cytoplasm. At the same time, it transports glutamate from cytoplasm into the mitochondria. That glutamate is one that's going to be involved in the formation of N-acetylglutamate. Now, this aspartate that's coming to the outside in the cytoplasm is going to combine with citrulline. So you can see citrulline is coming out, aspartate is coming here. They're going to combine to form what we call adeninosuccinate. The enzyme here is called adeninosuccinate synthase 1. So this enzyme is catalyzing the reaction between aspartate and citrulline to form adeninosuccinate. Later on, adeninosuccinate is going to be converted to arginine. And this reaction, it involves the removal of fumarate. So we know where fumarate can go. Fumarate can go to TCA cycle. So what's going to happen is that this enzyme, adeninosuccinate lyase, is going to catalyze this reaction, which involves the removal of fumarate, and this fumarate will go to TCA cycle, while arginine is going to continue in this same cycle, which is the urea cycle. Now, we are going to convert arginine to ornithine, and this reaction involves the addition of water molecule. So we are going to add a water molecule here, which hasn't been shown, and then we are going to produce urea. This urea will be excreted by the kidneys through urine, and this ornithine is going to be reused. So this is the urea cycle in a nutshell. Thank you for watching.